Now we're here at gene expression. So expression of genetics. Well, we talked about transcription translation. That's how you express your genes. But there are some nuances here because you're not expressing all your genes all the time. So let's talk about let's do a little vocabulary review. Your genotype is your genetic code, and your phenotype is the physical expression of that code. Transcription is translation. Transcription and translation is how you get from genotype to phenotype, right? But genotype and phenotype are not always equal. You don't always express all your genes. So, for example, diploid organisms, which are organisms that engage in sexual reproduction, because remember, diploid means you have two sets of genes, one from each parent. Diploid organisms can carry unexpressed genes, right? You have recessive and dominant. You might remember that. So you can have a masked gene. You can inherit an allele for blue eyes from your mom and an allele for brown eyes from your dad. Your eyes are going to be brown. You're still carrying the genes for blue eyes, but they're masked by the genes for brown eyes, right? But you can still pass on the blue eye genes to your offspring, even though you're not expressing it yourself. So that's one way genotype and phenotype don't equal. For diploid organisms, you can have recessive genes masked by dominant genes. So the genes are there, but they're not expressed. But more to the point, for bacteria, some genes are silenced while some are turned on. And that's another way genotype doesn't equal phenotype. That's not just true for bacteria, that's true for all organisms, but that, bacteria are not diploid. So this is the only, so we're really going to focus on that point in bold. So not all enzymes are produced all the time. The environment affects whether genes are expressed or not. There's this nature versus nurture debate, and it's stupid. It's so stupid because you cannot separate the two. There is not an organism on this planet whose genes are unaffected by the environment. The DNA code is not affected by the environment other than mutagens. But an organism turns genes on or off depending on environmental signals. So the environment tells the DNA which genes are you going to express, which ones are you going to keep quiet. And that changes dynamically, moment to moment, generation to generation. You can, especially for eukaryotes like us, it's not just the genes can be turned on or off. They can be up and down regulated. They can be made louder. They can be made quieter. If your genes were all expressed the exact same way all the time, regardless of environmental signals, you would never have a different mood. You would never do a different thing, right? So when you experience stress, what do you make? Stress hormones. How do you make stress hormones sometimes and not at other times? The environment sends signals to your DNA, and certain genes are upregulated or downregulated depending on the signals of the environment. Okay, so that is what we're going to talk about now. The environment affects how the DNA code is expressed. And this is also why different cells inside your body look different. All the cells in your body, sperm and egg excluded. Sperm and eggs, they're their own thing. That's sexual reproduction. But all the other cells in your body are genetic clones of all the other cells. Your neurons have the exact same DNA as your white blood cells, which have the exact same DNA as your bone marrow cells, which have the exact same DNA as your muscle cells, which have the exact same DNA as your liver cells. Why do they all look and act so different if they all have the exact same DNA? That is because certain genes are turned off and other genes are turned on. Stem cells, so when sperm meets egg in your mother's womb, that is the first cell of your body. And all the other cells in your body are clones of that first fused cell between sperm and egg. That cell, once sperm meets egg and they mix their DNA, that is the genetic code for the rest of your body, for the rest of your life. That cell divides into 2 clones, into 4 clones, into 8 clones, into 16 clones. At the beginning, those cells are not differentiated. The on-off position of the switches hasn't been set yet. But eventually you get cell differentiation. Some cells start to become muscle cells. Some cells start to differentiate into heart tissue. How does that happen? Well, depending on where the cell is in that developing, first it's a blastocyst, which has no differentiated tissue, and then it's an embryo, right, and then a fetus. Well, there's these chemical signals between the mother and the cells with each other that give signals for which genes to turn on and which to turn off. That's what we call cell differentiation. At some point, the heart cells will turn on the heart cell genes and will turn off the liver and skin cell genes. At some point, the skin cell genes will turn on the skin cell genes and they'll turn off the blood cell and the muscle cell genes, right? So, and then once that the on-off switches are set, that all the cell lines from that one cell are going to inherit the same on-off switches. A skin cell is not going to revert back to a liver cell. Skin cells have the genes to be a liver cell, but they're not, the switches are set on-off permanently in that case, okay? So this is why, so gene expression accounts for why blood cells, skin cells, and neurons all have the exact same genes. They're clones of each other, but they look very different. It also accounts for why bacteria forming biofilms look and behave very differently from the same species of bacteria that's not involved in a biofilm. Genes get turned on or off. Okay. So first, usually what you thought of as genes before this, before this section, you thought all genes were constitutive genes. Constitutive genes are genes that are always turned on. They're always being expressed. They're always being transcribed and translated into protein. Transcribed to RNA, translated into protein. Not all genes are constitutive genes. Constitutive genes make up about 60 to 80% of genes. So somewhere, depends on the organism. It's still difficult for us to really estimate because we haven't figured it all out yet. But constitutive genes are the genes that are expressed the same way all the time, no matter the environment. And that makes up maybe 60-ish% of genes, maybe 80%, a conservative estimate. That still leaves a very large proportion of genes that are highly influenced by environmental signals. All right, repression. We're going to cover repression, induction, operons, and epigenetic controls. Operons are how bacteria specifically control their gene expression. Epigenetics, that's a control method that's used by both us and bacteria. Repression is when you turn a gene off. That makes sense, right? So if you're repressing something, you're dampening it, you're turning it off. Repressible genes, this is the confusing part. Repressible genes are genes that are by default on, and certain environmental stimuli can turn them off. So a repressible gene is a gene that's already on, but you are able to turn it off. Repressible, it's able to be turned off, okay? So that's the counterintuitive part, because when you hear a repressible gene, you think it's off already. It's not off already. It's on, but you can turn it off. It's possible to repress it. It's able to repress it. Okay, so repression is turning off a gene. A repressible gene is by default on, but certain environmental stimuli will turn it off. Induction. Induction is when you turn a gene on. You induce it. Inducible genes are by default off, and certain environmental stimuli will turn it on. All right, so operons. This is how bacteria turn their genes on or off. They do it with operons. And different textbooks will have different ways of divvying up an operon because it has all these different parts. So this is the DNA. The DNA is color-coded by region. So there's several different, there's structural genes and there's regulatory sequences. There's all these different components. Different textbooks will divvy these features up in different ways. And there's no right or wrong way, but you need to know my way just so we can be consistent across the class. So I want you to know 3 components of the operon. There is a promoter sequence. So this is the operon here to here. And this is regulatory sequences out here that are not part of the operon, but they're related to the operon. So within the boundaries of the operon, there are 3 components. There's a promoter sequence. Promoter sequence is where RNA polymerase is going to bind. I mentioned it in the transcription translation lectures. RNA polymerase scans DNA for a promoter sequence because the promoter sequence is this long sequence. It's always the same. It always precedes the genes. There's a lot of junk DNA, right? The promoter sequence tells RNA polymerase, which is going to transcribe the DNA into the RNA photocopy. Promoter sequence tells RNA polymerase, this is the start. Here's the start of the gene. So here, RNA polymerase binds here. Operator. The operator sequence is where is where a repressor can bind. Sorry, I lost the word. It was on the tip of my tongue. A repressor protein can bind to the operator. A repressor protein blocks transcription. It stops RNA polymerase from transcribing. When a repressor is here, no transcription happens, no gene expression happens. The gene is off. When the repressor is removed, RNA polymerase can transcribe the genes. The gene is turned on. It's being expressed. Then you have structural genes. That's part 3. So part 1, promoter. Part 2, operator. Part 3, structural genes. And sometimes there's only one structural gene. Sometimes there's 3 related genes that are all adjacent to each other. Here there's 3 genes. They're all related to the same process there, so they're all linked up together. Structural genes is the protein blueprint. This is what's going to be transcribed into messenger RNA. The messenger RNA is going to the ribosome, and it's going to be translated into protein. The structural gene is the blueprint for the protein. Okay? This just controls whether that blueprint even gets expressed or not, right? So these are part of regulatory sequences. These are the actual structural genes. So promoter, RNA polymerase binds here. Operator controls whether RNA polymerase can transcribe the gene or not. So that's the on-off switch. And the structural genes, that's the blueprint for the protein. Okay. Now, there's this regulatory region that's not part of the operon. Not part of the operon. This is another promoter sequence. RNA polymerase can bind here. This is a structural gene for a repressor. Structural gene for the repressor. So this, because I told you that the repressor, the on-off switch for these genes, it's a protein, which means its blueprint is in the DNA, because DNA to RNA to protein, right? So to make this protein, you have to have a genetic code for it. The genetic code is here. So here's the genetic code for the repressor. So promoter is where RNA polymerase binds. RNA polymerase binds to this promoter, transcribes into messenger RNA the blueprint for the repressor. The messenger RNA goes to a ribosome. Ribosome translates it into protein, and the protein is the final product that can bind to the operator and turn off these genes. So you'll notice there's no on-off switch, there's no operator here. This is a constitutive gene. It's just produced all the time. These are not constitutive genes. Whether the repressor is bound or not is going to determine whether these genes are on or off. And the environmental signals, there's going to be signals from the environment that is going to allow the repressor to bind or unbind. Okay. Now, inducible genes, inducible operons, are operons that are by default off but can be turned on. That means by default, the repressor is bound here. Environmental signals will lift the repressor and allow these genes to be turned on and transcribed, translated. A repressible operon is an operon that is by default on. By default, there is no repressor here. By default, these genes are being transcribed, translated, but certain environmental signals will add a repressor. So under certain circumstances, the genes can be turned off or repressed. All right, now let's talk about the lac operon. The lac operon is an operon that encodes, so it is an operon that you find in E. coli. The lac operon controls the expression of lactose digesting enzymes. Lac for lactose. Lactose is a milk sugar. If someone's lactose intolerant, they can't digest milk sugar, right? So E. coli have these genes to digest milk sugar, and there's 3 enzymes here. There's structural gene for beta-galactosidase, beta-galactoside, beta-galactoside transacetylase. It doesn't matter. You don't need to memorize these. All you need to know is that lactose digestion occurs in a series of steps. Each step is conducted by a different enzyme. The 3 enzymes responsible for lactose digestion are next to each other on the E. coli genome. Now, this is an inducible operon. it doesn't mean all, I always have students, I go over this example and they think all operons are inducible. They think this is the only operon out there. I'm just giving you one example of an operon. There's all kinds of operons. This is one example. This one happens to be inducible, which means by default it is off, because E. coli is not usually digesting milk sugar. It's usually digesting other things. So what a waste of energy it would be to produce enzymes to digest milk when there's no milk around. That's just a waste of materials, it's a waste of energy, it's a waste. So most of the time, these genes are off. That means most of the time there is a repressor bound here. Now, in the presence of milk, when lactose is around, lactose, a derivative of lactose, binds to the repressor and removes it. So the very presence of milk causes, lactose is going to bind to the repressor, remove it. Promoter, now RNA polymerase, the way is clear, the roadblock is removed. RNA polymerase can transcribe these genes. Their transcripts are going to go to the ribosome for translation, and these enzymes that digest the milk sugar are going to be produced. When lactose is all digested and there's no more lactose around, the repressor is released, it binds back to the operator again, this whole expression process shut down. Okay? So only in the presence of milk sugar are these milk digesting enzymes produced. When milk is absent, the genes are turned off. Okay, so that is one example of an operon.